This lecture will serve as an introduction to confocal microscopy. So why confocal? Why is this worth a lecture? Why is it worth your time to learn about this? So another way of stating this question is, what problem does a confocal solve? So to understand what problem a confocal solves, it's useful to refer back to a more basic kind of microscopy and to use that as a reference for what a confocal does differently. So the kind of microscopy that I'm going to compare confocal to is called wide field fluorescence microscopy. And in this kind of microscopy, what we have is a sample. That sample has fluorophores inside of it. And these fluorophores are illuminated from light that comes from a lamp and goes through lenses and other things. And that light is projected onto the sample. And some of the fluorophores in the sample might be in the focal plane, as the ones you can see there on the dotted line are, and others might be out of the focal plane. When the light from the fluorophores that are in the focal plane gets focused back through the lenses and other stuff, that light is focused sharply onto a sensitive area detector, which is typically a camera. So the light from this fluorophore might be focused there, whereas the light from this other fluorophore might be focused there. In contrast, light from fluorophores that are outside of the focal plane, like this one, is blurred. The image of it is blurred when it is projected onto the sensitive area detector. And all of these things are happening in parallel. So we have a combination of sharp images of things that are in the focal plane and blurry images of things that are not in the focal plane. This problem of having out of focus objects that are contributing blurred objects into the image is what a confocal solves. So as an example, we have here a 15 micron kidney section image with the 63x 1.4 NA objective, and on the left you see the image taken with a wide field microscope, and on the right you see the image taken with a confocal. Another example are neurons in culture looked at with a 63x 1.4 numerical aperture objective, and you see on the left the image taken with a wide field microscope, and on the right you see the image of the exact same region taken with a confocal microscope. And I'll note that in the image on the right with a confocal, there are less blurry things. So this is a much more crisp image where images of things that are blurry and appear to be out of focus on the left-hand image have been removed on the one on the right, leading to enhanced contrast. So how does a confocal do this? How does it seemingly reject the light from things that are not in the focal plane, leading to a crisper image with less out of focus blur? I'm going to diagram the very basics of how a laser scanning confocal microscope works and compare it to the wide field fluorescence microscope that we were talking about just recently. So again, I'm going to schematize a sample with fluorophores both in the focal plane and out of the focal plane. I've colored them differently, but they are the same kind of fluorophore. I've just colored them differently because that will make the ray tracing easier to understand. So the way I've colored them is I have ones in the focal plane, which is that dotted line, in that red color, and the ones out of the focal plane in that purple color. So in a confocal, unlike in a wide field fluorescence microscope, the light source is typically a laser, and it goes through lenses and scanners and other stuff. And unlike in the wide field fluorescence microscope, where there's a very broad illumination, the light gets focused down to a very tight spot in the focal plane. So it converges into the focal plane into a really tight spot, the dimensions of which are given by the characteristics of the lenses involved, and then the light diverges outside of that focal plane. Now, what happens to the resulting fluorescence when these fluorophores are excited? So we're going to do a simple ray tracing diagram for fluorophores both in and out of the focal plane. For fluorophores in the focal plane, the light is aligned in a way such that when it goes back through the lenses and the scanners and other stuff, it is projected into a less sensitive spot detector. This detector is not a camera. You can think of it as a black box where light goes in, stuff happens, and a number comes out. But critically, before the light makes it to that spot detector, there is something called a pinhole in front of it. This is just an iris with a little hole. In a confocal, this pinhole is placed at a location and has a diameter such that light from things that are in focus, so in the focal plane, makes it through the pinhole more or less unimpeded into the detector, whereas light that comes from things that are out of focus mostly hits the edges of the pinhole and is therefore removed. So therefore, what we accomplish with this is to remove the light from things that are from objects that are outside of the focal plane. And furthermore, obviously, we don't just look at one point in the sample. We want to detect an entire plane in the sample, and the way we do that is by detecting in series or scanning the laser. So we point the laser to different locations in the sample, um, and therefore we generate the image in series as opposed to in parallel, which is what happens on the wide field fluorescence microscope. So the laser scanning is actually two-dimensional. So I showed in the previous slide how this laser scanned in one dimension, but it actually scans in two, and it does this raster pattern back and forth until it covers an entire plane. Once this is complete, you can do three-dimensional imaging by combining multiple two-dimensional planes. So we scan the laser in two dimensions, we move the sample in the z dimension, which is the focal axis, and then we repeat the measurement. And so we can get a three-dimensional data set. So here's an example. We can combine 2D planes to allow 3D imaging of, for example, a pollen grain. So on the right, you see serial optical sections taken with a confocal of a grain of pollen. And we've done what I mentioned before, but now we can see the result. We've scanned the laser in two dimensions. We've moved the sample in the z dimension, and we've repeated this over and over. And we can take all of these images and reconstruct that pollen grain in three dimensions. So clearly, something that is critical for all of this to work is the pinhole. This is the critical element for removing the out of focus light. If we remove the pinhole from the system, we would not have this critical property of increasing contrast by removing out of focus light. So an important question is how we set the pinhole size. And so there are two extremes. If we close the pinhole, we will reject more out of focus light and we will have higher resolution. Whereas if we open the pinhole, we will have more light, so more light will reach the detector, but we will have a resulting thicker optical slice or section. So you can imagine when you use a confocal, you are looking at samples, and with these sort of optical tricks that I mentioned in the previous slides, it's as if you're taking your sample and cutting it into sections. But you're not physically cutting them, you're cutting them with this optical trick. And because of that, this process is called optical sectioning, and we talk about the thickness of the slice as a parameter that we can consider. And so here are examples of the pinhole set to different diameters. So the pinhole diameter can be measured in different ways. One is in just microns, but one way that we measure it, which is comparable across confocals, is through something called airy units. And so the bigger the airy unit size, what you'll see is that the image looks blurrier. So if on the top right you look, you see that this is a 5 airy unit confocal pinhole diameter. The image looks sort of like a wide field image where there's a little bit of fuzziness as things above and below the focal plane contribute out of focus blur. Whereas the images of things with 0.5 area units or 1 look much crisper. So here is an example of an experiment that was done to illustrate the same phenomenon and to try and find kind of to illustrate what the optimal recommended setting is. So these are images taken with different pinhole sizes, and then all the other parameters were adjusted to get a similar brightness of the image. And they measured then two things. The slice thickness reported by the system, which is a measure of resolution, that is the curve in blue. So you can see the bigger the pinhole size, the bigger the slice thickness. And in red, they measured the relative laser power needed to get an image of equivalent brightness. And so what you see is that as you decrease the pinhole size, the slice thickness gets smaller, therefore the resolution gets better, and the relative laser power goes up. So you need more light to generate an image of a similar brightness because you then reject a lot of the resulting fluorescence light at the closed pinhole. And so the generally accepted practice in the field is to use a pinhole diameter of around 1 ARE unit because that is a good compromise between these two opposing forces, more resolution versus more light. When you have more than one fluorophore, there are different conventions, and if you train on a confocal at MSL, you will see that what we recommend is to set it to 1 ARE unit for your longest wavelength fluorophore. So confocals can image multiple fluorophores. So let me briefly explain how this works. So this is a quite complicated diagram, and I'm going to strip it down to the most essential things you need to understand for how a confocal can image more than one fluorophore in a very simple confocal. So these are the elements that you need to keep in mind, and I will walk through them by exciting a sample and collecting the emission fluorescence from that sample. So if we imagine that we have an excitation laser that hits a polychroic that reflects that laser but allows other light through, then the laser goes to scanning mirrors which move and uh, bounce the laser off an additional mirror that's fixed and then scan the sample. The resulting fluorescence is emitted in all directions, but some portion of it goes back into the objective and it goes right back through the light path, but now it goes through the polychroic. The reason being the polychroic is transparent to light that has wavelengths different from the laser. It bounces off another mirror. It goes through the pinhole, and once through the pinhole, it enters a little detection box where there are various dichroics and filters. And so the first dichroic reflects the green light, which goes through a final emission filter in front of one of 3 photomultiplier tubes, which are one of these spot detectors where the light goes in. So this is done either for one frame or for one line in the sample. And then we can easily switch to another laser, which in this case I've in the diagram made green, and we collect orange fluorescence that as it goes back goes through the first dichroic, because that dichroic is transparent to this light. It transmits all this light, but then bounces off the second dichroic and goes through an emission filter into the second photomultiplier tube. And then finally, we can excite the sample with orange light, get the resulting red fluorescence, and that goes through both dichroics and through an emission filter into the third photomultiplier tube. And usually we do this sequentially. We don't do all the lasers at once because we don't want crosstalk. We don't want the fluorescence emission from one fluorophore to be confused and end up in another photomultiplier tube. And so we usually cycle through them. So we will do this one, this one, this one, and then repeat it. And that way we form an image where each channel is acquired independently and we don't have any light, or we minimize rather the chance of light leaking from one fluorophore into our detection of another. Now, this is a very simplified schematic. Modern confocals have flexible wavelength detection windows. So this is an example with a Zeiss LSM980. This is not actually a very modern confocal, but the way that this does the flexible wavelength detection is actually very similar to more recent Zeiss models. And so since I liked the schematic, this is the one I'm using. So the important parts here are the same. So what you can see in this case is the light will go, let me just get a laser pointer. The light from the lasers, these are the lasers, it goes into the sample through here, and then the fluorescence light comes back. Element 8 is the pinhole. And then don't worry about everything that's happening here, but the light bounces off this element. Eventually it gets here. That's not super important how it gets here. Just trust me that it does. And this element is something called a diffraction grating, which when you illuminate the diffraction grating with light with multiple wavelengths, it separates out those multiple wavelengths into spatial positions. And once you have transformed the spectral separation into a spatial separation, you can use the fact that they are spatially separated to direct them with various prisms to the 3 detectors here, which are this thing that says 13 and these 2 that say 12 and 14. And so by adjusting the angles of these things, one can make windows where different portions of the emission fluorescence are directed to different detectors. And if you're interested, there's a nice video here that you can take a look at as well, where this is animated, or the version of this for the 980 is animated. Now, different companies do this in different ways. The Zeiss LSM980 is very similar to the 710 in that it has a diffraction grating to do it. Olympus uses a technology called the volume phase hologram. Leica uses prisms, and Nikon has many options to do the same thing. So all the companies do this to some greater or lesser extent, and they all have different mechanisms to do it. But the idea is the same. In modern confocals, you're able to a large extent, steer the light that you want to the different detectors that you have on the system. A very nice feature of many modern laser scanning confocals is they can do bright field at the same time. So this is a schematic of a Leica SP8 inverted confocal. You can see the objective here, a slide here. So all of this is what we talked about before, the lasers and that put the light into the sample. And then here you can see the light, the fluorescence coming back and getting split into different detectors. But I want to draw your attention to this. There's an additional detector on the far side of the sample that collects the laser light that goes through the sample. And that allows us to create a transmitted light image of the sample, essentially for free. It's only just one more data channel, but it doesn't come really with a compromise for speed or sensitivity. And we acquire that bright field image at the same time as we acquire the fluorescence, and this can be quite useful. So confocals are great, but they do have problems. So let's talk about some of those problems. So first of all, we are throwing away some of the light, hopefully mostly out of focus light at the pinhole, and we are detecting therefore only some of the light on a spot detector that's less sensitive than a camera. So this is a problem because we are losing photons and not making the most of the ones we get. So this is not a very sensitive technique. Another problem with laser scanning confocal is that the scanning process, because we acquire images in series going point by point, is slow. So it takes us longer to take an equivalent image than some other technique. And finally, we use the laser to illuminate the sample, and lasers are usually high power sources of light, so this can be very damaging to both the fluorophores and the biological integrity of any live samples that we might be looking at. So let's see what we can do about sensitivity. So how can we increase the sensitivity of a laser scanning confocal? So the problem, just to state it more explicitly, is that standard photomultiplier tubes, this is one of the problems, are noisy and not very sensitive. The way a PMT works is that a photon hits a sensitive material, an electron is released, that electron is amplified in a chain reaction, an electrical signal is measured at the end of that chain and is converted into a number. This process is noisy and it's also not very efficient. So it has a quantum efficiency for a classical photomultiplier tube that maxes out at around 30% at a wavelength of 400. But even at wavelengths between 500 and 600, the efficiency is around 20%. So we're losing most of the photons. We are not detecting most of the photons that hit the detector. The quantum efficiency means if a photon hits the detector, what's the probability that I will detect it at the other end? And you can see it's quite low for the photons we care about. So what can we do about this? So the solution are better detectors that cost a lot of money. And so there's different versions of these. There are GASP photomultiplier tubes that many companies sell. The GASP stands for gallium arsenic phosphorus, and so they're made out of a material that is more sensitive on most of the wavelengths that we care about. You can see that quantum efficiency of the GASPs in red compared to the normal PMTs that are typically called multi-alkali in blue. Other companies, specifically Leica, use hybrid detectors, which are a combination of a PMT and an avalanche photodiode. Don't worry about what that means or exactly how it works. Just know that those detectors are similarly higher in efficiency than the sort of standard multi-alkali ones, as you can see in that graph, where the signal to noise ratio for those detectors in red is compared to regular PMTs in blue. And finally, Olympus has recently come out with a detectors called Silver, which have a sensitivity similar to the GASP, but they have significantly lower noise. So again, different companies make different kinds of better detectors. And that is one solution to this problem. You throw money at it, you get a better detector, you collect, you manage to make more of the photons that you collect. So another problem with the sensitivity of a laser scanning confocal is that the pinhole blocks the light. So mostly it blocks light from things that are out of focus, but that light still has information that we could use. So if you imagine, again, the schematic, if you have the pinhole open to 1 area unit versus 5 area units, and you take an image of a 100 nanometer bead, the light that comes from the one on the left did come from that bead, and it can be used to gather information about that bead. So discarding that light decreases the sensitivity of laser scanning confocal. And so a possible solution to this is to rethink this whole pinhole and point detection scheme by throwing money at it. And so two companies have come up with array detectors. For Zeiss, this is called AriScan. For Nikon, this is called N-Spark. And the idea is that instead of projecting the light back to uh, one photomultiplier tube, we project it back to an array of detectors. Here they're in this sort of hexagonal honeycomb pattern. Here they're in more of a square pattern. So the idea is we're capturing more light, and furthermore, we can do some clever reassignment of the light based on where it ends up to simultaneously increase the resolution. So this is a nice way of getting around this issue that the pinhole blocks some of the light and therefore decreases the sensitivity. So another problem with laser scanning confocal is that it's slow. You can see that, just to be reminded, is because scanners are involved that have to raster the image, the laser through the sample, and that's a slow process. So what can we do to increase the speed of a laser scanning confocal? And just for reference, sort of a typical image on a typical confocal, though this can vary a lot, but again, typical takes around 1 second. So what can we do about this? So the first is that solving one problem can often solve more than one problem. So increasing the sensitivity allows higher speed with the same signal to noise ratio. If we make more out of the photons that the sample gives us, we can go faster and not lose any quality. So in other cases, we can make trade-offs. We can scan less, so we can trade field of view for speed. So for example, if the only thing we care about in the field of view is in the middle and only occupying 10% of the field of view, we can crop so that we only scan there, and therefore we can go much faster. An extreme version of this is to just crop a line scan where we only scan a line, and we can make chimegraphs of things like calcium transients and voltage at very, very high frame rates, including up to around a kilohertz. So that's an extreme version of this trading field of view for speed. Another option is to scan coarsely. So we trade resolution for speed by scanning with fewer pixels. And this is something that we'll discuss in a little bit more detail later in this lecture. Another option is to use resonance scanners. These are expensive. They are scanners that resonate. That's why they call resonance scanners. And this resonance is at a high speed. They're typically imaging at 30 frames per second or higher. These are mirrors that oscillate at a fixed high frequency. They have less flexibility, so they can't rotate and they can't zoom like normal scanners usually. And they often have issues with uneven illumination. But they are an option to go faster. That said, beware, because the speed, if you want to go faster, this will cost you photons because you will collect fewer from each location. So just the fact that you can go faster doesn't mean that you will get enough photons to do anything useful with that additional speed. And so this is something to really keep in mind if you're using a resonance scanner. Finally, a problem with the laser scanning confocal is that it uses a laser, and therefore this can be very damaging. And when we talk about damage, we're talking about how light can damage both cells and the fluorophores that we put inside of those cells to study different things. And so fluorophores can be irreversibly destroyed by light, and this is a phenomenon called bleaching. And live cells can be severely damaged by the light to the point where they have blebbing, or their physiological processes break down or slow down. Things like cell division don't take place, migration gets slowed down. And so we term this sort of typically photo damage. So what can we do? Again, solving one problem can help you solve another. If we increase sensitivity, that will allow us to have less damage because we will be able to put in less light, knowing that we more efficiently use the light that comes out as fluorescence, as emission photons. So we preserve our signal to noise ratio with less light put in. So again, solving one problem helps with others. We can also scan coarsely. We can trade resolution for less damage. So we don't go to as many locations, and this allows us to not damage the sample as much. We'll see this in a little bit more detail later. We can also scan faster. So if we don't spend as much time on each pixel, we will trade sensitivity for less damage. So we won't collect as many photons. We'll have a noisier image, but we will damage our sample less. One other very useful approach to minimizing damage is to use longer wavelengths. This is particularly useful for live cells. Longer wavelengths have lower energy and are less damaging to live samples. So a typical example of this is if you are trying to track nuclei and you compare using hoaxed, which is a UV excitable dye, versus silicon rhodamine hoax, which is a version of it that's been shifted into the far red. The latter is much less damaging. And so highly recommended to use that one instead of the hoaxed. The final thing is you can improve the sample for either live or fixed samples. You can use brighter, more stable fluorophores. Fluorophore development is something that has made a tremendous strides in the last few decades, and so the kinds of fluorescent proteins that we have available to us now are dramatically better than the ones that we had, 20 years ago. And the same is true for uh, fluorescent dyes as well. And so it's worth your time to look for new and improved fluorophores. If you are using fixed samples or looking at fixed samples, you can also use mounting media. So you place those samples in special media that has anti-bleaching properties. And so that can also help you reduce damage that the laser scanning confocal does to your fluorophores. Whatever you do, you will never be able to escape from what is sort of colloquially called the pyramid of frustration. You are trying to optimize for several things at once. You want high spatial resolution, perhaps. You want high contrast. You want high temporal resolution. You want good specimen health if it's alive. And you're trying to figure out how to navigate through all this because the different things that you're trying to optimize for are pulling you in different directions. And so in the rest of a good chunk of the talk, we're going to talk about how we navigate all these competing constraints. So what parameters can we adjust on a laser scanning confocal? For contrast, we can adjust the laser power, something called the averaging, and the scan speed. For spatial resolution, we can adjust the zoom, the image size, the frame size, or the pixel size. So I will explain what all of these terms are, how we adjust them, and why we might want to do one thing or another in the coming slides. But before I do that, let's just have a small sidebar to say that the contrast and spatial resolution are not actually trivial to define and measure. So let me just illustrate these concepts with an example that will hopefully give you a sense of what it is that we're trying to do. So what you see here is an image of an Argolite HM slide. This is a slide that you can buy that has constant non-bleachable patterns with systematic spacings between 700 and 100 nanometers. And the reason you don't see that very clearly is this is an image that I've taken with low resolution. It has very big pixels relative to the features in the object, and low contrast. You can see it's quite noisy. This is a version of the same image with high spatial resolution and still low contrast. So it's still noisy, but you can now tell that there is a decreasing separation between these two patterns from here to here. And that goes from 700 nanometer spacing to 100 nanometer spacing. Now, I have also taken a version of these in high resolution, in high contrast with low spatial resolution, and with high contrast and high resolution. So you can see how these concepts of contrast and resolution interact in this sample. And hopefully that's helpful when you're trying to think about what these things mean and what you are trying to accomplish. So to visualize contrast, One way of thinking about it is by drawing a horizontal line all throughout one line in this pattern, which is supposedly exactly the same intensity. And so you can see that I've done this for the image with low contrast and for the image with high contrast and plotted the resulting intensities in these graphs, and you can see those are very different. So you can, a measurement that will reflect contrast, or a possible measurement that will reflect contrast, is to measure the signal defined as the delta between the intensity where you have a line and where you don't. So that's what I've marked in that green there. And the noise, which we can take as some measure of the standard deviation of the signal where we have the bar. So once we have those two numbers, we can divide one by the other, and that can be called a signal to noise ratio. So that is one way. There are others of measuring the contrast, and that allows us to compare these two samples and capture this thing that we see visually, which is that the one on the right, there's some measure of quality that's clearly better than the one on the left. For resolution, we can uh, visualize this by plotting intensity along vertical lines in this same kind of sample. And so the idea here is if you plot vertical lines, you're trying to see where you can tell the difference between these two patterns that are close to each other, which show up here as these two peaks. And so what you can see is, as a possible measure of resolution, you can estimate the peak-to-peak distance of the smallest clearly identifiable spacing. And so you can see here, you can barely tell them, but you can see that there are two peaks, whereas here you can only see that in this in this pattern, which has a larger spacing. So the image on the bottom has a higher resolution. Okay, so let's talk about contrast. And so the first obvious parameter that we can use to adjust the contrast is the laser power. So here's the effect of the laser power by going from 0.01% all the way up to 1.28% in factors of 2. So I've gone from 0.01 to 0.02 to 0.04. And I'll note an important thing is I've measured that on the system on which I did this, which was our LSM900 Tonks, the laser power is perfectly linear with this setting. So it is actually true that 0.02%, which is the thing that it says in the software, is actually twice as much laser power delivered to the sample as 0.01%. So you see the result of adjusting that parameter on the multiple images that I've taken here. And there's an obvious thing, which is the more laser we apply to an image, the brighter it is. Now this, the way I've displayed them on all of them have the same display settings, is actually not a very useful way of displaying them if I want to study their contrast. Instead, it's better to auto scale the display of all the images without changing the data so that I can see them at a similar visual brightness. When I do that, it becomes quickly apparent that the quality of the images increases as I increase the laser power. As can be seen by the fact that the one on the top left looks, that neuron looks very noisy compared to everything's around it, that's around it. Whereas on the bottom right, it's a nice crisp image, and I can see small processes that are lost in the noise in the image in the top left. Another way of capturing the same thing is to draw a line through the image and to plot the intensities across that line. And so that's shown here. And again, it captures the same idea where if you look at the image on the top left, it's very noisy, whereas the image on the bottom right, the peaks, which represent the various processes of that neuron, are much more crisp and better defined relative to the minimal noise in the rest of the sample, as shown by the fact that these areas are much, much flatter compared to this. So that is laser power. When you see this, one natural question is to say, why not increase the laser power to the maximum? And the reason is that there are diminishing returns. Improvements get smaller with higher laser power. So you can increase the laser power, but the visual improvements that you see or that you can measure get smaller as you increase the laser power by more and more. And this is expected from just some basic considerations of how we're detecting the light, which I won't go into here, but this is a very typical result. So if you get a very small improvement by doubling the laser power, perhaps it's not worth it to double the laser power. Another reason to not increase the laser power to the maximum is the possibility of signal saturation. The detectors on a confocal have a limit to how many photons they can measure reliably. If you increase the laser power so much that too many fluorescence emission photons come out such that they breach that detector limit, then you will be left with an inaccurate measurement of the intensity in your image, as well as the potential for distorted morphology, as I will show you later. Another reason you might not want to increase the laser power to the maximum is bleaching. More laser power can make the signal fade as the fluorophores are destroyed irreversibly. Finally, there's a phenomenon called fluorophore saturation, which is that more laser power does not always lead to more emitted photons. And the reason is at a certain point, you're exciting every molecule in the sample into the excited state. They're decaying and emitting photons, and you keep repeating this over and over. So putting in more light actually doesn't do anything for you. It does not lead to more emitted photons. And so what you want are emitted photons to improve your signal to noise. If you're not getting any, there's no point in doing the increase in laser power if you've reached that point. So let's talk a little bit more about signal saturation. Be aware of this. This is a very common mistake when people are setting up a confocal. Saturation will irreversibly destroy your data, meaning if you've done this and saturated your data, there's no going back. There's nothing I can do for you to recover the thing that you lost. And so even if you're not interested in measuring anything, saturation will distort the morphology. If you compare the level of detail on the left and on the right image, I've increased the laser power and shown the pixels that are saturated in red. All those pixels that are saturated in red have the same intensity. So things that are actually filamentous show up as big blobs because of this phenomenon of saturation. So again, even if you're not really interested in measuring intensities because your experiment does not require that, these giant saturated blobs clearly have distorted the morphology of the thing that you are interested in. The other reason that you don't want to saturate your data, your images, is that it makes quantification basically impossible. So if I now draw a line through the same feature in the saturated and the non-saturated image, you can see that in the one that's okay, we have clear gradations in fluorescence intensity, whereas in the saturated one, we have these plateaus as the signal reaches the maximum possible, and then everything there just shows up as a flat identical number, and so we can't capture the real variation that is present there, and so we can't quantify properly. So how do we avoid saturation? So to avoid saturation, we want to find the brightest object in the brightest experimental condition, and set up our settings such that the maximum pixel intensities in that object are at half the full range or less. So for example, these are a bunch of DAPI nuclei. This one in the middle is very bright, so I've set it up there, and you can see that this is at sort of half the full range. So we don't need to be at half the full range. We just want that to be our maximum. And the reason is, sure, we could push it all the way almost to saturation, but then we don't have any safety margin. And usually having a 2x safety margin where if something is twice as bright as the thing you measured to try and set up these settings, that it would still not be saturated. So to summarize, if we look at the laser power, we can either be in a situation where we have low bleaching and low contrast or high bleaching and high contrast. So what are other things that we can adjust? So the two other things we can adjust are averaging and the scan speed. So averaging means that we scan lines in the sample multiple times and then average them together, or you can think of it as scanning the frame multiple times and averaging it together. So it's like you can think of it, we're taking multiple pictures and averaging them. The other option is to reduce the scan speed. So the laser dwells more at each location, therefore we get more photons out of each location. And so that is another way that we can adjust the contrast. So what are the effects of these three ways that we've discussed that we can adjust the contrast? So I'm going to compare the effect of scan speed versus laser power versus averaging. And I'm going to start by showing the same display settings in all the images. And so if we increase the laser power, the factor by which we have increased it is at the bottom. So factors of 2, 2, 4, 8, and 16. The image gets brighter, and we've seen before that it also improves in quality. If we do something similar by decreasing the scan speed or increasing the average at the same equivalent light dose, so 2x for the laser means we've doubled the laser, 2x for the scan speed means it's twice as slow, 2x for the averaging means we took two images and averaged them, and so on and so forth for the other light doses. You can see that decreasing the scan speed or increasing the averaging does not result in a brighter image. It is also the case that it's very hard to see from these images whether we've accomplished any improvements in quality. So I'm going to switch them to auto scaling, where they're auto scale so that the brightness is very similar. And you can see here that very clearly, as we increase the light dose through any of the three methods, the quality goes up. Another way of showing this is by drawing a line through the sort of same part of the sample, and we can see how noisy it is before we've done anything versus how much cleaner it is, albeit not perfect, when we have drawn the lines through the images with the highest light dose. So if we look at scan speed and averaging, uh, for scan speed, we can elect to have a low contrast image but high speed, or a high contrast image with low speed. And for averaging, if we do very little averaging, we will also have low contrast but a higher speed, or higher contrast with a lower speed. So we have all these options. How do we decide between them? So a few general rules of thumb that are useful. First, the same total light dose tends to lead to a similar quality. So if you double the laser or you double the averaging or you slow it down by a factor of 2, those typically lead to similar improvements in the image. Um, as far as speed goes, it's obvious that if you increase the averaging or reduce the scan speed, you will be slower. So in that regard, increasing the laser power has an advantage compared to the other two. In terms of total signal level, increasing the laser power can saturate the signal intensity or the fluorophore emission. So you have to be careful when you're increasing it that you don't do either of those things. For the fluorophore emission, what you want is if you double the laser power, you should see a doubling of the emission. If not, you have some nonlinearity, and it's advisable to back off in terms of increasing the laser power. Finally, there's bleaching. So for bleaching, there are reports in the literature that are sort of a little bit all over the place, and there's some sense that averaging is more gentle than slowing the speed, which in turn is more gentle than increasing the laser power. What I'll say is that that is probably true for the papers in which they measured it, for the fluorophores in which they measured it under those conditions. But bleaching is extremely complicated, and really the only way to know for sure what the bleaching behavior between the different options on a laser scanning confocal for improving the quality, how they compare, the only way to know for sure is to check it for yourself in your sample with your fluorophores, with your conditions. So what about the photomultiplier tube gain? So this is a very prominent parameter in a confocal. It's something you can tweak. It's something that um, I myself recommended that people tweak in the past. Why have I not said anything about it yet? So the reason is that on the particular laser scanning confocals that I have in my core now, which are basically two Zeiss LSM980 with gas detectors, here is what happens if you change the photomultiplier tube gain. These are images shown on the same scale, and there are 4 voltages that I've applied to the photomultiplier tubes. And you can see that as you increase those voltages, the images get brighter. But the key question that we want, these are all on the same scale, is whether the images increase in quality. So let me auto scale them to the same brightness, and you see here that there isn't really a significant difference in quality between all of the images. And I've measured this in more careful, systematic ways, and I've never managed to see any difference whatsoever. In fact, if you just do one small other thing, which is just draw a line through them, you can see that there's really not that much difference. And so if you're in a situation like this, where increasing the photomultiplier tube gain increases the brightness but doesn't increase the quality, um, and has, so again, no effect on contrast, but a large effect on signal intensities. The recommendation on these systems and on systems that behave similarly is to leave the PMT gain at the minimum, which in this case is 500 volts for these gas detectors. And the justification is that this will give you more room to increase the laser power without signal saturation. If you just make the image brighter, but don't get any quality in return, you're wasting your dynamic range. You're precluding increasing the laser power more to get a better quality image. So that is not recommended, and therefore we recommend instead to leave the PMT gain at a minimum, which in these confocals means 500. This is something that you'll have to check on your own system if you have something different to see whether it's worth it to change this parameter or not. So all of the things I mentioned so far are ways of optimizing the contrast on a laser scanning confocal. So how do we optimize the spatial resolution on a laser scanning confocal? So a critical question is, what level of biological detail do you need? And beware, because more is not always the right answer. So you may not need high resolution if, for example, you're studying nuclei. So if you look at the image on the left, it is much more pixelated than the image on the right. But if you are just counting nuclei and the nuclei have on average this spacing, it is good enough for counting nuclei. The image on the right is overkill, and you will waste time acquiring an image with details that you do not need. On the other hand, if you're studying mitochondria, the image on the left is a disaster. You can't do anything with it. The image on the right is better, but it's still quite bad. And so you probably need to do even more than what we have done on the image on the right. And so if the answer to this question of what level of biological detail you need is as much as possible, let's talk about what your approach should be. So there are a few key concepts that you need to be aware of to understand um, how you decide what pixel size and what spacing in the Z dimension you want to use to take both 2D and three-dimensional data sets. So the first key concept is that in any microscope, point objects will have a blurred image. And that blur will be roughly on the order of 200 nanometers in the XY dimension and roughly 500 nanometers in the Z dimension. So that's one key concept. Images of point objects like single molecules, which can be thought of approximately as a point object, for our purposes, are blurred at a significantly larger scale than the size of the object. So just for reference, for example, a molecule of GFP, the diameter of that is roughly 2 nanometers. It looks actually like a can, not a sphere, but that is much smaller than the 200 nanometers of blurring in XY and the 500 nanometers of blurring in the Z dimension. So that's one key concept. Another key concept is that you can think that there's a minimal distance to distinguish two-point objects called resolution. And so in XY, the way this shows up is as follows. If you have point objects, each of the images of those point objects will be a blurry circle with additional rings because of a phenomenon called diffraction or airy patterns, as you can see them here. And if we zoom in, there's a minimal distance where we can tell that two of these objects are apart. If they're closer together than that, the blurriness merges in a way that we're not sure actually if we have two objects or one. There's a similar version of this same phenomenon in Z, where if we have two objects that are far enough apart, we can say with confidence that they are two objects. If they're very close, we don't know actually if we have two or one. And there's this some sort of sweet spot in the middle where we can just barely tell that they are two objects, and that is the number that we call the resolution in Z. And these two numbers, the resolution in XY and the resolution in Z, depend on the fluorophore wavelength and the objective numerical aperture. So another key concept that we have to understand is that of optimal sampling, both in the XY dimensions and in Z. And so if we look now at what pixel size would be appropriate to capture all these details, the answer is clearly not this. So this pixel size is very big. What this means, if this were the size of the pixel, is that all of the information that we're seeing here would get collapsed into a single number from this entire spatial region. And so that obviously loses a lot of the detail. Pixels of this size, by contrast, really don't get us any extra information. We're just getting a magnified view of the blurriness that's there. So there's sort of an optimal pixel size, which is roughly taken to be about a third of the resolution in XY, according to something called the Nyquist criterion, which is a more general idea about how you should sample things that have certain spatial scales. And so this is roughly a pixel size that will get you all the information without under or over sampling. There's a version of this in Z, which is what spacing, if we're going to take a Z stack, should we use between the slices? And so if we want maximal resolution, and that resolution is that red bar seen there, clearly the spacing that I've shown here as an example is too big. We're going to lose a lot of details that we could access. On the contrary, this kind of spacing is much too small. We're just going to magnify the blur, take longer, and photo damage our sample. But somewhere in the middle, there's an optimal, which again is roughly going to be the resolution in Z divided by 3 according to the Nyquist criterion. So this tells us that one of the really important things is to set the Z spacing between the slices, which is not that difficult, but also to set the pixel size correctly. This is a little bit trickier, and so let's discuss how we set the pixel size correctly. And so we're going to go back to the parameters that we can adjust on a laser scanning confocal. And for resolution, the critical parameters are going to be the zoom, the image size, the frame size, and the pixel size. So how do we set the pixel size correctly? So you can imagine that we have a sample. That sample interacts with the microscope, and we create an image with pixels that represent different locations in that sample. So what are these 4 things that I mentioned? So image size is not the size of the image in megabytes. It's the size of the sample, the area of the sample represented by the image in microns. So this image represents an area in the sample that is x microns times y microns. The frame size is the number of pixels in x and in y in the image. The pixel size is therefore obtained by dividing the image size by the frame size, and it is the size represented in the sample by 1 pixel. So it's x sub I microns times y sub I microns. Finally, the zoom is the scan area of the laser. If that number is big, it means the laser is scanning over a smaller area of the sample. If that number is low, it means it's scanning over a bigger area in the sample. And the zoom directly determines the image size. If we zoom in, the image size will be smaller. If we zoom out, the image size will be bigger. The 2 things that are under our direct control are the zoom and the frame size. The others respond to what we do with the zoom and frame size according to the relationships I've discussed so far. So let's do an example. Let's say we have an image that looks like this, and we decide to increase the frame size by making the pixels smaller. If we do that, we will have higher resolution, but we'll also have slower imaging because the laser needs to go to all these locations, and we will have more bleaching. If we instead take the zoom and increase it, we will have, again, higher resolution, but it will only image a smaller area with more bleaching because we'll have a higher concentration of laser per unit area, but we will have this smaller pixel size that gives us that higher resolution. So what is the recommended workflow? The recommended workflow is first, determine the pixel size that you need. And this decision is going to be based on the size and spacing of whatever structures you're interested in. So if you're interested in nuclei, you've got to get one answer. If you're interested in the microstructure of mitochondria, you're going to get a very different answer. Second, determine the image size that you need. So this is going to be given by the size and spacing of your experimental unit. If you need to have at least 1 cell, because that is your experimental unit, you need to have an image size of at least 1 cell. If you need to have a cluster of cells, because you're studying junctions between them, for example, you need to have an image size that can fit multiple cells. Similarly, if you have a tissue structure that you're interested in, you're going to need images that fit that structure that you're interested in. So tissue structures, cells, organelles, whatever the thing you're interested in that is going to determine the image size that you need. Once you know what image size you need, set the zoom to get the desired image size. Then set the frame size to get the desired pixel size. Frequently in many confocals, this is a button that you can just press if you want the highest possible resolution, and it'll pop the number right out. And then finally adjust other parameters to balance the contrast, the bleaching, and the speed. So an important thing to know is that there's more to confocal than laser scanning. So laser scanning confocal is a variant of confocal microscopy that optimizes for spatial resolution and contrast. But there's another common variant called spinning disk confocal, which I'll talk about briefly, which is more geared towards preserving specimen health and temporal resolution. And so how does this work? I realize this is a very complicated diagram. I recommend you go to the video I've noted there. The idea is you have an array of laser beams hitting the sample, each with its own pinhole. And then you move this array to scan the entire sample, and you project the resulting fluorescence onto a camera, which is a very sensitive type of detector. So instead of having a raster pattern like this, you can have a sort of a spinning disk scan pattern like the one on the right, and this can allow you really fantastic imaging. So this is an example from Sarah Cohen's lab where she's looking at the dynamics of peroxisomes in red and mitochondria in green, and we were able to take full Z-stacks at high resolution every 5 seconds for at least 30 minutes without any problems. So you can see all sorts of interesting behaviors. Let me do that, run that again. Um, Okay, it's not going to run again. You can scroll back in the video if you want. There are all sorts of interesting behaviors you can see in that video: mitochondria fusing, splitting apart, peroxisomes wandering around near them, et cetera, et cetera. And so that's the kind of thing you can do with a spinning disk confocal. It's very well suited to this kind of experiment. Oh, there we go. We did get it again. So you can see, mitochondria uh, splitting, fusing before, moving around, all sorts of interesting stuff going on. Okay, so one of the problems with spinning disk confocal is that you have multiple pinholes, and so if you have a thin sample, that's not a problem. But if you have a thick sample with a lot of fluorescence, what can sometimes happen is light from things that are out of focus can leak through adjacent pinholes and lead to something called pinhole crosstalk. So you're not actually rejecting all of the out of focus light as efficiently. There are solutions to this. You can make a larger distance between the pinholes, but none of these solutions are perfect. And so there's really no free lunch. The pinhole crosstalk reduces axial resolution. And so it doesn't have as good axial resolution as a laser scanning confocal. But it is nevertheless a very useful system that you may want to consider for the things that you're doing. Confocals are really workhorse instruments in biology, and the reason is this parameter space that they can explore intersects with a lot of interesting biological samples. So confocal is great, but it's not perfect. And so let me explain some of the things that are challenges when you're using a confocal. So first, thicker things can be very hard to image. And so why? So the reason is often because of a phenomenon called light scattering, which is if you have a material that has a lot of heterogeneity in refractive index in its components, and they're all mixed together, this degrades transparency. So if you shoot a laser through such a material, it doesn't propagate ballistically as you would expect if it were perfect, but rather has this sort of more diffusive quality as it goes through the sample. And so that makes it very hard to get to be able to create a crisp image of whatever's in that sample. So what are your options? Well, one option is to image thin things. If thick things are a problem, we can image thin things like cells cultured on surfaces. We can take thicker things, cut them into slices that are thin and image those. Um, we can image also transparent things, either naturally transparent, like C. elegans, which part of the reason it was picked as a model organism is that it is transparent, or chemically cleared. We can use various chemicals to render tissues transparent by removing things that have different refractive indices and then creating, in the end, a solution that has the same refractive index as whatever's left, and that usually makes things transparent. Or finally, we can use infrared light in a type of microscopy called multiphoton. So this is one challenge. Yes, confocal is better at imaging thicker things than something like a wide field fluorescence microscope, but um, things much thicker can cause problems even if you have a confocal. Another challenge inherent in confocal is that visualizing and interpreting three-dimensional data, which is what you often get, can be really hard. So this is an example of the neuron. And so on the left, we have the entire data set, which are 62 images. And on the right, we have it in movie form. This is kind of a neuron that I've been showing you over and over um, in this presentation. And you can see that there's a lot going on here. So how do we visualize this and how do we interpret it? And so there's different options and you'll have to choose between them. So one option is to look at a single Z slice at a time. That's the image on the left. Another option is to do what's called a maximum Z projection. So in this case, we summarize all of the images with one image, where the rule to create that one image is to replace every pixel in that image with whatever the brightest version of that pixel was in the entire Z stack. So it's like a collapsed version of everything, and it can be a useful way of summarizing. Um, However, you can see on the image on the left where I've done this maximum Z projection with a parameter called gamma at 1, which is the typical default. It's very hard to see both bright things and dim things at the same time. So the soma is very bright and we can't see it very well, and we're also having difficulty seeing maybe some of the dimmer things. In that case, you may want to explore reducing the gamma or increasing it. It depends on what software you use, what direction you need to go in. So in Fiji, it would be decreasing it. In Imaris, it would be increasing it to bring up the brightness of the dim things like the small processes, while not letting the brightness of the bright things change as much. And so this allows for us to visualize this a little bit more easily. I have an Imaris viewer video where I explain exactly how to do this. You might want to check that out. Um, Another option is if you want to capture the fact that some things are above or below some other things, you might want to do a depth projection where you color the resulting projection with colors that tell you what height things are in the sample. And we're going to focus on this thing here so that you can take a look at how that might play out in an example where one thing is going above the other thing. So let's zoom in there. So this is a maximum Z projection with gamma 1 or gamma 0.5. And you can see there's clearly things going in different directions. It's unclear if they're connected, if one is above the other, which one. Here, there are individual Z slices where you can see clearly one of the thick thing is above the others. And then here we have this depth projection where they clearly have different colors. And so that helps you understand that one of these things is below the other. You can also take advantage of things called orthogonal views. So in orthogonal views, what you can see is different planes in the sample. So you can imagine the sample as this cube in X, Y, and Z of data, and you have an XY image. That's the typical one that you look at. That's the thing that's also in the middle. You have an XZ view, which is from the side, and a ZY view, which is from the other side. And so these are these views. The XY is in the middle, the XZ is on the bottom in this case, and the ZY is on the other side. And here you can see that we're in a particular Z plane, which is this one, and we can see this object, so that fiber. If we go, so this allows us to see it without the visual clutter of where exactly we're cutting it, which is what these lines mean. But if I go to another plane, we're now going through this object, and you can see this is above the other one. So this can be a very useful way of visualizing three-dimensional data. You can also visualize it completely in 3D, and there are different options for rendering, which may or may not be more useful to study the things that you're interested in. You can also decide whether to deconvolve the data or not to deconvolve it. So deconvolution is a procedure that improves contrast and resolution, and we have it available at MSL. So these are examples of the raw data that has been maximum projected, or deconvolved data that has been maximum projected. And you can see the difference in quality. And if we zoom in here and look in XY and in XZ, you can see the improvements in terms of reduction of noise and a slight increase, particularly visible in the Z resolution. So this is something that you might want to consider. So to sum up, I would say that in biological microscopy, laser scanning confocal is like a Swiss Army knife. It's very useful for many things. So I have one of these knives at home. I use it for all sorts of stuff, but it's not the best tool for everything. So if I need to use a saw, I will not use this. I will use a proper saw that I have in my tool shed. Nevertheless, it's very useful to have this instrument. It allows you to do a lot of things, and that's why we have so many in biological research and why there's such a useful tool for so many things. And a major goal of the training that you'll receive at MSL on a laser scanning confocal and other instruments is to help you navigate this pyramid of frustration where you try to adjust parameters to balance spatial resolution, contrast, temporal resolution, and specimen health to be able to answer the questions that you care about in your sample.